Hi, welcome back to the world of bioinformatics. One of the hallmarks of modern research is the generation of enormous amount of raw sequence data. Thus, the very first challenge is to store and handle the staggering volume of information through the establishment and use of computer databases. The development of database to handle the vast amount of molecular biology data is thus a fundamental task of bioinformatics. So today we will discuss some basic concepts related to biological databases. A database can be defined as a computerized archive used to store and organize data in such a way that information can be retrieved easily via a variety of search criteria. Databases are composed of computer hardware and softwares for data management. Although data retrieval is the main purpose of all databases, biological databases often have a higher level of requirement, known as knowledge discovery, which refers to the identification of connections between pieces of information that were not known when the information was first entered. For example, databases containing new sequence information can perform extra computational tasks to identify sequence homology or conserved motifs. These features facilitate the discovery of new biological insights from raw data. There are several reasons to search databases. For instance, when obtaining a new DNA sequence, one needs to know whether it has already been deposited in the databases fully or partially, or whether they contain any homologous sequences. Moreover, some of the databases contain annotations which has already been added to a specific sequence. Finding annotation for the search sequence or its homologous sequence can facilitate its research. Or to find some similar non-coding DNA structures in the databases, for instance repeat elements or regulatory sequences. Other uses for specific purpose like locating a force priming sites for a set of PCR oligonucleotides. To facilitate the access and retrieval of data, sophisticated computer software programs for organizing, searching and accessing data have been developed, and they are called data management systems. These systems contain not only raw data records but also operational instructions which help to identify hidden connections among data records. Coming to the classification of biological databases, biological databases are developed for diverse purpose, encompass various types of data at heterogeneous coverage, and are curated at different levels with different methods, so that there are accordingly several different criteria applicable to biological database classification. According to the scope of data coverage, biological databases can be classified as comprehensive and specialized databases. Comprehensive databases cover different types of data from numerous species, and a typical example is GenBank. On the other hand, specialized databases are those that cater to a particular research interest, for example Flybase, HIV sequence database and ribosomal database project. These are databases that specialize in a particular organism or a particular type of research. According to the level of data curation, biological databases can roughly fall into primary databases and secondary or derivative databases. Primary databases contain original biological data. They are archives of raw sequence or structural data submitted by the scientific community. GenBank and PDB are examples of primary databases. Secondary databases contain computationally processed or manually curated information based on original information from the primary databases. A prominent example of secondary database is SwissProt, which provides a detailed sequence annotation that includes structure, function and protein family assignment. According to the types of data managed in different databases, the biological database can roughly fall into the following categories, and the most popular databases are biomolecular sequence database, biomolecular structural database, genomic databases, database for small molecules, database of biomedical literature, metabolic pathway databases, database for specific organisms, and so on. First one is the sequence databases. It can again divided into nucleotide sequence database and protein sequence database. There are three major public sequence databases that store raw nucleic acid sequence data produced and submitted by researchers worldwide. They are GenBank, the European Molecular Biology Laboratory Database, and the DNA Data Bank of Japan, that is DDBJ, which are all freely available on the internet. The GenBank database is perhaps the best known nucleotide sequence database available at the NCBI. The European counterpart of GenBank is the EMBL, that is European Molecular Biology Laboratory Nucleotide Sequence Database from the European Bioinformatics Institute EBI. Another primary nucleotide sequence database is the DNA database of Japan, which is operated by the Center for Information Biology in Japan and is the primary nucleotide sequence database for Asia. The three databases operators NCBI, EBI and CIB comprise the International Nucleotide Sequence Data Collaboration and synchronize their databases every 24 hours. This means that by connecting to any one of the three databases, one should have access to the same nucleotide sequence data. Although the three databases all contain the same sets of raw data, each of the individual databases has a slightly different kind of format to represent data. Next is the protein sequence databases. Margaret Dayoff was the inventor of Protein Information Resource, that is PIR, in the 1960s at the National Biomedical Research Foundation for investigation of evolutionary relationship among proteins. Analysis tools for protein databases are provided by PIR, which are freely available to the scientists. In 2002, the Protein Information Resource and its worldwide partners EBI and Swiss Institute of Bioinformatics were granted an award for the National Institute of Health to make a universal protein resource, that is UniProt, by merging the databases of PIR, PSD, SwissProt and translated EMBL. Next is the structural databases, and the most common examples are PDB, molecular modeling database, vector alignment search tool, conserved domain database, nucleic acid structure database, and so on. And from the above databases, now we will discuss PDB and NBD in detail. PDB, that is Protein Data Bank, is a source for the three-dimensional structural data of huge biological molecules, includes proteins and nucleic acids. It was established in 1971 by the research collaborators for Structural Bioinformatics, RCSB. The data submitted by scientists from different parts of the world are easily available through the internet. The PDB is supervised by the Worldwide Protein Data Bank, and this database archives atomic coordinates of macromolecules determined by X-ray crystallography and NMR. It uses a flat file format to represent protein name, authors, experimental details, secondary structure, cofactors and atomic coordinates. The web interface of PDB also provides viewing tools for simple image manipulation. Next is the Nucleic Acid Structure Database, NBD. This database provides us 3D structures of nucleic acids. The NBD contains information about experimentally determined nucleic acids and complex assemblies. NBD can be used to perform searches based on annotation relating to sequence, structure and function, and to download, analyze and learn about nucleic acids. Next is literature databases, which provides the library of life science work done all over the world. There are various literature databases like Medline, Sites Explorer, Patent Abstracts, etc. Next is the pathway databases. To comprehend molecular interactions and chemical reaction networks, the pathway database is used by pathway marks. Various pathway databases are available like KEC, MANET database and so on. Next is the chemical database, which is a collection of chemical information precisely planned, and the best examples are PubChem, Zinc, Drug Bank and so on. Next is enzyme databases. They cover an extensive range of properties and functions such as structure, occurrence, kinetics of enzyme catalyzed reactions and metabolic functions. There are so many examples for enzyme databases like Brenda, Ripis and so on. Next is disease databases. The disease database provides all disease related informations. It is a cross-referenced index of diseases, symptoms, medications, signs and so on. And the best example is OMIM. Next is domain database, which is a database for ancient domains and full-length proteins. Example CDT, that is conserved domain database. And the genomic databases are collections of genome sequences of many species. It interprets and examines them and provides the free public access. Example is mitochondrial DNA database, mouse genome informatics, plant genome project maintained by National Science Foundation and so on. Here we discuss a collection of biological databases, but by no means this collection pictures the whole range of databases available nowadays. Thank you so much for watching this video. Please share and subscribe.